Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Abdul Rhafiq bin Mahzan and my metric number is 190006. Okay, so today I would like to present about my internship program. Okay, I done my internship, Alhamdulillah, at Malaysia Nuclear Agency for the duration of 24 weeks from 8 March 2021 and until 20 August 2021. So, okay, I will introduce about background of Malaysian Nuclear Agency, just a short brief. So the Malaysian Nuclear Agency was located at Bangi, Selangor. At 1994, the name was the Malaysia Institute of Nuclear Technology, means until 2005, the cabinet was approved the change of name. The name was changed to Malaysia Nuclear Agency. Okay, this is my department, as you can see in the red box. This is red box. Okay, my department was located at block number 23. So my department was named Radiochemistry and Environmental Laboratory. My department provide the services, which is unit under Waste and Environmental Technology Division, responsible in developing radioactivity analysis techniques in environment and food samples. Okay, this is my mini project topic, special distribution of heavy metal, stable isotope, and soil erosion using fallout radionuclide FRNs at Kuala Selangor Nature Park, Selangor. Okay, for the introduction, the quantification method for soil erosion assessment using radionuclide analysis of fallout radionuclide is proposed to correlate with the special distribution of the heavy metal and stable isotope burial and mangrove sediment. The use of chemical fertilizer and the burning of petroleum may also be linked to anthropogenic heavy metal production in the setting. The research question is, what is the concentration of selected heavy metal concentration in sediment of Selangor River, Malaysia? Now, okay, for the research objective, to understand the movement of stable isotope carbon between component of wetland aquatic ecosystem by using carbon isotopes and related techniques. For the methodology, the chosen study area is Kuala Selangor, one of the northeast district of Selangor, which is located 60 km northwest of the city of Kuala Lumpur. The chosen area still has several patches of upland forest, swamp forest, and some mangrove. So this is a sampling stations. As you can see, there are 3 pictures. 1, 2 at the near with Selangor River, and also 3, 4, and 5. Sorry, 3 and 4. The station was located in the Kuala Selangor Nature Park, and so on. So as you can see, 10, 11, and 12 was located nearby with paddy field, coconut field, and palm oil plantation. Okay, so this is the coordinates. Okay, so the experimental approach, how we are taking the samples sediment and soil sediment, soil sample. So the sampling method using the coral, sediment trap, and biomass. As you can see, this is the coral. This material called coral. For the type of sample we taken, soil, sediment, water, and plant. For the plant, we take the mangrove leaf. For soil sample, we are taken 20 to 30 centimeter depth core of soil. So for the sediment, 40 to 50 centimeter depth of core. So every sample we taken will be slashed every 2 cm. So which mean every slash of the sediment core, 50 cm sediment core will be sliced every each 2 cm so we can get the chronology of the sediment, every sediment. Okay, so this is the lab work, sediment sample analysis flow and soil sample. First of all, when we take the sample into the lab, we weigh the wet sample. We get the weight we get the wet weight. Okay, then after we get the wet weight, we place the sample into the oven at 60 degrees Celsius until the sample become dry. Until the sample, the weight of sample becomes stable. After that, we re-weigh the dried samples. Okay, after we get the dried sample weight, we crush and grind the sample, sediment and soil sample by using rock lab machines until the sample become fine. Fine soil sample will be filtered using 2 millimeter sieve. Okay. Then there are there are 2 sample divided. First sample for FRN sample and the second sample for NAA sample. For FRN sample, the sample that filtered by sieve will be filled into well-type container and compressed for 21 days. Okay. For NAA samples, we filtered the sample and fill into a vial tube. After that, we can get the sample into the machine. For the FRN, for the gamma, we put into the gamma spectrometry. For the NAA sample, I send the sample at the another department. So this is for the result and discussion. Okay, I'm sorry that I can't get the results because of challenge due to this pandemic. So I can manage to finish the project because of I can get the pelepasan untuk masuk ke office untuk menghabiskan saya punya projek. Okay. So for the discussion is gamma spectrometry system is used to measure the activity of radionuclides based on the energy and intensity of the gamma radiation emitted from those radionuclides. Every gamma emitting has its own characteristic energy which can be used for its identification, and the intensity at such energy enables the activity concentration of the radionuclide in a sample to be quantified. Among the radionuclide that can be measured using the system are cesium-134, cesium-137, radium-226, and radium-228, and potassium-40. Okay, that's all from my mini project. So last but not least, I would like to thanks to my supervisor, university supervisor, Dr. Muhammad Amir Hamzah and Dr. Muhaimin. Also, big thanks to my supervisor at company, Puan Nurul Ashikin, for big for guidance and instruction that given to me. Okay, that's all from me. Thank you. Assalamualaikum warahmatullahi wabarakatuh.